Okay, so let's get started. As I said, my name's Helen, and today I'm giving the webinar for the Ensembl release 85, which came out last week. It says underneath my name here that I'm part of the Ensembl outreach team, and what that means is that we're a team of 4 people, and there's me on the left. Next to me is Emily, our team leader, and then we've got Ben and Victoria. Emily is with me today on the webinar, so if you have any questions throughout the webinar, if you look in the software, there's a box to the right, and at the bottom of that there's a chat box. So if you have any questions as we're going along, Emily's sitting at her computer ready to answer any of those, and if there are any outstanding at the end of the webinar, I'll see if I can answer them myself. Okay, so what do we do in Outreach at Ensembl? We do workshops, so we travel around the world and we deliver training on Ensembl at different institutions. We also do video tutorials and then webinars like this, and I'll mention some tutorials that we've done recently that you might find really helpful. We also try and keep up to date with the help documentation on the website. That's a really useful resource if you've got any questions as you're using it. And we also have email support, so if you can't find your answer in the help documentation, we welcome all of your emails and we try and solve your problems for you. Okay, so the webinar today, I'm going to try and keep it fairly short. I'm just going to give a brief introduction to Ensembl. So today I'm imagining you've got some experience with the Ensembl website. If you don't, then I will point you to a place where you can learn a lot more about it. I'm going to then show you a little bit about the release cycle, why we have one and what it means. And then I'm going to give you the highlights of what came out of the latest release, which was Ensembl 85. And with that, we had some data updates, and we've also added some web features. And here's just a little bit more about the data updates and the web features that came out. So we have new epigenomes for our regulatory build, and we also have new gene sets for human, mouse, and rat. We've added some CRISPR data, so you can see a track with CRISPR sites added. And we've also, for chimpanzee, imported some new symbol names. So some of the genes in chimpanzee have been renamed. For web features, we've got a new tree viewer from Wasabi, and I'll show you a little bit of that. And I'm going to show you some of our new track highlighting options as well. So we'll have a demo for that. So my very brief introduction to Ensembl. First of all, Ensembl is a genome browser, but why do we need them? Very briefly, the first genome that was sequenced was just 5 kilobases. In 2004, the human genome sequence was released, and that was 3 gigabases. So each genome itself is a lot of data. Since then, we're creating a lot more genomes and duplicate genomes. So very recently, the 1,000 Genomes project was completed. So we're getting a lot of genome data, and this is what genome data looks like. So it's pretty meaningless without further annotation. So that's part of what Ensembl does, and Ensembl is one of the is a genome browser, and there are lots of others. These are the 2 other main ones. So there's UCSC and NCBI Ensembl. As well. So I work for Ensembl, but people use different genome browsers depending on their preference. And what do genome browsers do? Well, what does Ensembl do? First of all is we import assemblies. So sequencing data is assembled into genomes, and we bring those in from public archives. So we don't do anything, we don't assemble any genomes ourselves. What we do is we annotate those genomes. So from the raw sequence, we use biological data to determine where genes are. We also get variant data and add that to the genomes. The regulatory features, we get regulatory data, and then we annotate the genomes with those. And then based on all of the genomes that we have in Ensembl, we do comparative genomics with all of those species. So in addition to annotating those genomes, we provide a way to display and export that data. So the display is the browser, so the website as you'll see it. And then exporting data, we have an FTP site, we have a Perl API and a REST API. And we also have a tool for exporting data. So if we move down the line to tools, we have Biomark, which is a really handy way of exporting data. Other tools we have is that you can upload your own data and view that in the browser. We also have the variant effect predictor, BLAST and BLAT. So if you want to find out any more about that, I would definitely show you where you can find out more information later on. So in Ensembl, we have 80 species, which includes our pre-sites. We focus on vertebrates, although you'll notice in the red circle we have some non-vertebrates. These are important model organisms that we've used, and they're really useful comparative genomic outgroups. It was launched in 1999, and we use Ensembl gene annotation and Havana gene annotation. And as I talk a bit later about the gene sets that we've got, I'll tell a little bit more about the Ensembl and Havana. And Ensembl is a project that's from the EBI and the Wellcome Trust Sanger Institute. So that was my very brief introduction. Now we're going to move on to the release cycle. So in March 2016, we released Ensembl 84, and we took a bit longer this time, but generally our release cycle is 2 to 3 months. And what happens in that release cycle, as you'll see appearing on the circle, is we import new data and do lots of different things. First of all, if there are any new genome assemblies that we're going to add to Ensembl, that's the first thing that we do. If we're updating the software, then we'll do that. And then we update gene sets on any of the species that we already have. And of course, if we import a new genome, we'll have a new gene set for that. Then we run our comparative genomics pipelines. If we update any of the variation data, so that's data that we bring in from variation archives, and any regulation data that we're bringing in, you'll see we've got some of that this time. And then any of the web features, so new or updated interfaces, so that's things to do with the browser. So this, in general, takes 2 to 3 months, and so you'll see that we need a release cycle just to add all of this information and to keep the website stable for you. So if you're wondering what release we're currently on and where you can find out what that means, if you go to ensembl.org, you can see there's a news box in the top right-hand side. It's here in this red box, and this has got some of the main features that came out, and it tells you that this is release 85, and it came out in July 2016. If you want to read full details about all of the data that we added, you can click inside that full details button there. The reason we work on a release cycle rather than just drip feeding information is that if you're working on a certain data set and then you don't do any work for a while and you come back maybe a few years later, up to 3 years, you may want to work on exactly the same data. As I mentioned, we import new data, so if you want to work on exactly the same thing that you were working on before, then you'll need to use one of our archive sites. And so we keep archive sites for up to 3 years, so if you wanted to work on exactly the same data, you could go to the E85 archive site. So when we release E86, we'll freeze this website. So E85 will stay as E85 for the next 3 years, and you can always come back and work with this exact same data. Okay, so first of all, this is the first thing that I'm going to talk about today, is the regulation data that we've brought into this release. So we've imported 30 new human epigenomes from the Roadmap Epigenomics project, and just small, it's quite small, you might not be able to see this, but I've provided a PDF of these slides, and I'm also, I hope, recording this webinar. Let me just double check that before we carry on. Yes, I'm recording this webinar, so tomorrow I will also be able to update this, I'll upload this video to YouTube, so if you want to look back at any of this stuff or listen to me all over again, then you can do that on YouTube. Anyway, so yes, here are the 30 new human tissues that we've brought from Roadmap Epigenomics, and we've also brought in 34 human tissues from GTEx EQ2QTL, but that's growing soon. So what is Roadmap Epigenomics, and what is GTEx EQ2L? So Roadmap Epigenomics is a consortium that's trying to produce a public resource of human epigenome data to further biology and disease research. They're using next-gen sequencing technologies, including DNA to study DNA methylation, histone modifications, chromatin assembly, and small RNA transcripts. And to do that, they're using stem cells and primary ex vivo tissues. So this is normal tissues, healthy tissues. If you want to find out more about that project, I've put the URL at the bottom. So the other project we've brought regulation data in from on this release is GTEx-EQTL. So GTEx is the Genotype Tissue Expression Project. And again, they're obviously trying to provide the scientific community with a resource. And here they're using multiple human tissues from donors who have already had their genomes densely genotyped to assess genetic variation within the genomes. And they're using RNA expression data here. So again, if you want to find more about this project that we're bringing data from, you can look at the URL that I've put at the bottom of this slide. All right, so if you're in Ensembl, how do you find this data? So I've just jumped us straight to a location, a region detail page here. So depending on what gene you're interested in or what region of the genome you're interested in, you can jump to anywhere in the human genome. And then if you click on this cog here to configure the page, you'll get this box jump up. And then we're in the regulatory features here. And we've got, this is just a screenshot, but we've got a list of all of the different tissues and cell types that we've got. And this data has just been slotted in with all of the other tissues that we already had. So you can just select any of these. I've got some pre-selected in my screenshot. And then they'll show up as tracks. As standard in our in this region in detail view, we have the regulatory build track, and I've turned on the left ventricle, the lung, placenta, and thymus tissues that were brought in for this release, and you can just see the different activities that they have. Each one of these blocks is a regulatory feature. I've cut off the regulatory legend, but red means that it's a promoter. And if you click on any of these promoter boxes or regulatory feature boxes, you'll get a pop-up and it will tell you the ID for that feature and a bit more detail about it. So this one is a promoter. So if you were to click on this stable ID, it would bring you to the regulation tab. So this is a new tab that opens at the top. This was the location tab. This is the gene that I searched for, and this is the regulation tab for that. So here in the regulation tab, we have this summary, and this is where you get the features displayed. So this regulatory feature was found in these tissues, but it may well have been found in other tissues too. So now that we've come to the to the feature that may have been found in any of these tissues from any of the projects, and you can see more tissues than we saw on the previous page listed down the side. If you want to find out where the data came from that we used, we'll click on source data, and that tells you that some of the Roadmap Epigenomics found this feature, as the Blueprint and the Encode projects. So you can click on any of these links and find out more about where the data came from. Okay. All right, so next for new data is that we've updated the gene sets for human, mouse, and rat. So for human and mouse, we've updated the Gencode set, so that's our gene set that we have. So we've got Ensembl annotation and the Havana manual annotation, and we've also updated the CCDS genes. And in addition to that, we've got rat, Ensembl and Havana gene set updates to that. So what is Ensembl and Havana annotation? Ensembl uses biological data, so cDNAs, mRNAs, to do a computerized pipeline. So we take this biological data, align that to the genome, and do automatic annotation. Havana do that manually, so they take biological data as well, but they have a person looking specifically at each piece of biological evidence and then aligning that to the genome to do gene annotation that way. These are the, Ensembl has automatic annotation for all of the species that we have in Ensembl, and where Havana have done any annotation, we pull that over too. And in the case of human and mouse, this is the Gencode gene set. So Gencode is the gene set, the merged gene set of the Ensembl annotated genes and the Havana manually annotated genes. And this is the default gene set used by lots of big projects. CCDS is an agreement between ourselves, the Sanger Institute, UCSC and NCBI, and that is the consensus coding DNA sequence set. So this is a gene set of only coding genes, so you don't get UTRs on the end, but it's just a gene set where 4 different groups have agreed that that transcript exists. So yeah, we've updated the Gencode gene set and the CCDSs for human and mouse, and we've also updated the gene set for rat. Okay, so if you're interested about your gene set, you want to know more about it, but you don't know where to find information, again, I'll welcome you to go to the Ensembl homepage and then find your species. And here, if we click on mouse, that takes you to the mouse homepage. And then we've got more information about this gene build. This way you can find out about the gene sets. And here's a little table with a summary of it. So you can see that the mouse, we had the first gene build was released in July 2012, but it was last updated in May 2016, and that came out in this release, so in July 2016. And this is the Gencode set, and that's Gencode M10. And if you want to find out more about the gene annotation processes, we've got links here as well. If you want to see what this data update means to the tracks, so we have a CCDS track that you can turn on. So again, if you configure the page, then you can find this under genes and transcripts. The Ensembl standard gene set is the Gencode one for human and mouse. So if you just click on genes, this will be the main track. And if you click on that and you find information, it tells you more about the Ensembl gene set, and that this is Gencode version 25 now. So this is just another way to find more information. Okay, so another piece of data that we've added to the site is that we've added some CRISPR sites. So the Wellcome Trust Sanger Institute have a genome editing team, and they very recently published a paper where they show that they've predicted CRISPR sites, and they've developed a database to help you design your genome editing experiments. And we've added the data from this database for human and mouse in Ensembl, so you can view the CRISPR sites there. Just last week, we released a blog post about this, and my colleague Ben. So if you go to ensembl.info, that will take you to our blog. If you use this link specifically, it'll take you straight to that blog post. And then this link at the bottom is for the genome editing, the paper that they published with this data. So if you want to see what that looks like in Ensembl, again, I've just gone to configure this page on the region in detail, and if you just search CRISPR in this search box up here, then you can turn on these CRISPR sites. So what does that look like? You can see here highlighted, we've got all of these green tick marks, and if you zoom in a lot more, you'll see that these tick marks are actually the CRISPR sites themselves. So in dark green, we've got the PAM motif, and then the empty box, we've got the gRNA binding sequence, or potential gRNA binding sequences. So hopefully you guys will be finding that useful. Okay, so the next piece of new data we've brought in are the VGNC symbol names. So we've just done this for chimpanzee for now. So I don't know if any of you are familiar with the Human Genome Nomenclature Committee. So that's a project that was set up to make human gene names consistent. So there's a unique gene name and a unique gene symbol, and that's they're sort of developed with assistance of the community, just to make sure that scientists are using consistent gene names and aren't making up their own ones which are different from what their competitors were making up. So it's just a way of getting unique standard gene names. But that was just for human. So as sort of an extension for that is the Vertebrate Genome Nomenclature Committee, or VGNC, and they're going to be responsible for assigning standardized names to other vertebrates which don't already have their own nomenclature committee, and then these names are going to be in line with the human homologs. So the VGNC has just released this for chimpanzee, so we've imported that. And what does that mean? That might mean that the gene that you're looking at in chimpanzee may have been renamed. So you can see this is the archive, so this is what an archive site looks like. So this was Ensembl 84, and you can see this chimpanzee gene here was called GTDC2, and then this is the Ensembl gene name. That has now been renamed to POM-GNT2, but the Ensembl stable ID is exactly the same. So if you were using the stable IDs, then you'll still be able to find your genes. And we also have this old name linked to the synonym. So this is the current Ensembl E85, and so this gene has now been renamed that, but the stable gene ID is exactly the same. Okay, so now moving on, that was quite quick. Now moving on to some of the new views that we've got in Ensembl. So this is the first one. So we've got a Wasabi tree viewer. So we were previously using GelView to show sequence alignments, and now we've moved on to using Wasabi. And this is just a web-based environment to look at the sequence alignment in a phylogenetic tree, and you can just do so in your browser, and it doesn't require anything to be installed. So Wasabi was released by a project in Northern Europe. I forget exactly which country, but if you want to read their paper, that's the link to the Wasabi paper. So in Ensembl, the way to get to this Wasabi thing is go to your gene, and then in the left-hand menu, click Gene Tree, and then that will bring up the gene tree for the gene that you're looking at. And then if you click on any of the nodes, so here I've clicked on this one, and this pop-up menu opened up. And then to view the subtree, I'll click on a view in Wasabi. And what that brings up is another pop-up with this Wasabi app inside. And this is really easy to navigate. You've got zoom buttons and slide buttons, and you can make each panel bigger and smaller and scroll along. You can also copy and paste this URL here to share just this with your colleagues if necessary. Yeah, this is a really neat way of looking at the alignment next to the phylogenetic tree. Okay, so the last thing that I'm going to talk about for new things in Ensembl is track highlighting, which hopefully you guys will find useful. So we've, I'm just going to do a demo here, but we've just briefly, when you add a new custom track, that will be highlighted initially until you roll your mouse over it. As you hover your mouse over any track, that will be temporarily highlighted. And we also have a little toggle button to turn on highlighting for any track, and that will remain highlighted until you untoggle that. So moving now onto the Ensembl browser, so just on any region in detail, if you want to add some custom data, I'm just going to paste in some data here and call it webinar, and that's bed data. So hopefully that will add fairly quickly. There we go. So that's uploaded. So this is some new data, and I'm going to go to the nearest region with that data so we can go and see it. Hopefully it'll load. Okay, yeah. So you can see here's the track I just loaded, and it's already highlighted in yellow. And as I hover over it, it stays yellow, but as I move away, it's now not yellow anymore. So that's just a way of seeing which tracks you've just added, in case you've added lots of tracks and you wondered which ones they were. This is a way to see that. And then additionally, I mentioned as you scroll your mouse over any of the tracks, you can see highlighting there. So if you're not sure the upper and lower bounds of any track, or if you're just trying to follow some data along, this highlighting is really useful. If you know you're definitely interested in a certain track, you can click on the name there, and then this icon here is to highlight or unhighlight. So you can see the pencil's drawing a black line, so that means there's no highlighting. As you turn it on, the pencil's drawing a yellow line, and you can see this track now is permanently highlighted regardless of where else I have my mouse. So that's the new highlighting options. And we welcome feedback on any of these things, so please do email help desk if you have any feedback. Okay, back to my presentation. Okay, so just to wrap up now then, if you ever need any help with Ensembl, please do email us. So that's me, Emily, Ben and Victoria. So we answer all the help desk questions. If it's something that we ourselves don't know, we work in the same office as the whole team, and we definitely do send a lot of questions their way, so we can answer them for you. So that's questions, bugs, any problems you're having, if you've got any feedback, anything else, we welcome. We would love to hear from you. We also have a public mailing list, so this tends to be for more technical questions, but we have dev@ensembl.org. If you want to hear more about our releases and anything new in Ensembl, then we have announce@ensembl.org as another mailing list. And as I mentioned briefly, I'm going to upload this video onto YouTube. So this is Ensembl Help Desk has a YouTube account, so you can find that. And then I've just got this again because it's a great place to find training. So I only really introduced you very quickly to Ensembl today, and I just jumped to screenshots that you may not have recognized. So if you want to learn a lot more about all of the things that we have in Ensembl, we have a lot of training videos, so short clips. If you want to learn more about Ensembl, so more of the details about where we get our data from, what we do with that data, and then how you can use that data, we very recently created this browser series. So that's a training course that's on the EBI Train Online. And so we have videos there. So the videos are half an hour to an hour long of data and presentation, 7 of them. So it'll take you more than a day to do all of this. So we have the video tutorials, and then alongside each video we have a few exercises you can do to try, and then we have the answers to go along with that. And I really do encourage you to take a look at that. It's really useful. So this is a picture of the team as of a year ago. We've had a few changes. I'm not in that picture. So we've had some new starters. Not very many people leave. So that's the team. That's all the people behind Ensembl. Please do follow us on Facebook and Twitter. And as I mentioned before, we do blog posts about some of the data. So the CRISPR stuff we've got on ensembl.info, so you can find details about that. And then I'd like to thank all of the Ensembl team for bringing all of these data in. They're all responsible for each release that we do. Everybody has a hand in that. And then I'd especially like to thank all of our funders for making Ensembl free and making it available for us all. Okay, I'm going to close my presentation down now and see if there have been any questions. Okay. I don't see any questions from anybody in this chat box, so I would like to thank you all for attending today. Hopefully you've learned a lot. If you have any questions that you didn't use the chat box for, please do email the help desk. Yeah, it would be great to hear from you. All right, thanks again. Have a nice rest of your day.